neurotransmitter action. Actually, I want to start with a little bit of a bigger picture, and then we'll look at the details of synaptic transmission. So basically, what we're talking about is synaptic transmission is a form of chemical signaling, and I want to put it in the larger context of other types of chemical signaling that occur. There are some similarities and some differences in the way that neurotransmitters work as compared with hormones and other signaling mechanisms. So we're going to start with this one and put it in context, and then we'll look at more specifically at the synapse itself. Okay, I'll pull this out.
Cells have a variety of ways of using chemicals to communicate with each other. An important means of intercellular communication in the nervous system is synaptic transmission, in which one neuron releases neurotransmitter molecules onto another. The neurotransmitter molecules bind to and activate receptors on the target cell. The activated receptors then activate target molecules within the cell. Other receptors act as ion channels that transduce the chemical signal into an electrical signal. Other well-characterized forms of chemical communication include paracrine signaling. In paracrine signaling, a cell releases signaling molecules that activate nearby target cells with appropriate receptors. Paracrine signaling acts over a longer distance than synaptic transmission. Another form of chemical communication, called endocrine signaling, refers to the secretion of hormones into the bloodstream. The hormones thus reach tissues throughout the body. Those cells with appropriate receptors become activated. Chemical signaling of any sort requires several components. A molecular signal that transmits information from one cell to another. A receptor molecule that transduces the information provided by the signal. And a target molecule that mediates the cellular response. The part of this process that takes place within the confines of the target cell is called intracellular signal transduction. Within the cell, the signal is typically amplified. Let's look at an example of signal transduction, beginning with a receptor protein that can bind to the neurotransmitter norepinephrine. The activation of a single receptor by norepinephrine can lead to the activation of numerous G proteins inside cells. This is the first amplification step. These activated proteins can bind to other signaling molecules, such as the enzyme adenylyl cyclase. In this way, adenylyl cyclase becomes activated. Each activated enzyme generates a large number of cyclic AMP molecules. This is the second amplification step. Cyclic AMP binds to and activates another family of enzymes, the protein kinases, that can phosphorylate many target proteins. While not every step in this signaling pathway involves amplification, overall, the cascade results in a tremendous increase in the potency of the initial signal.
Okay. Amplification is an important concept to understand. It's the way that a cell cell signal potency is increased, and there are several different molecular mechanisms for amplification. It becomes a really important thing to understand when we think about learning and memory. If a signal can be amplified, it could, forget it. If a signal can be amplified by some of the molecular mechanisms that are mentioned in the animation, then it can, in a sense, convey a firmer connection between two neurons, a more sort of hardwired connection between two neurons. So amplification is a potential mechanism of plasticity or change in the way the brain operates. And when we think about learning and memory, we have to think about mechanisms of plasticity or change, right? So the reason I wanted to kind of show this one first is it also presents this general scheme that we have to think about, and this is true throughout all of physiology, that the way that body cells communicate with one another is a signaling cell sends a message or signal which is received by a specific receptor that fits that signal in a lock and key mechanism. So these two molecules fit exactly together with one another. That receptor is target molecule is located on a target cell and elicits the response of the signal. Now, the signal can be a neurotransmitter, and that's the type of signal we'll be talking about, but a signal can also have an autocrine function. It can act on the cell that has released it, okay? Or it can have a paracrine function acting on a nearby cell in the same tissue. Yes?
Does paracrine usually denote separate that the whatever cell that is giving off neurotransmitters is communicating with several cells, or just that it's communicating with one cell that's not next to it?
It can be either. It can be communicating with just one cell or a number of cells, but they are close by. The principle by which the signal travels is simple diffusion, right? It's not being transported through the blood system. It's just diffusing.
So paracrine would
local.
So paracrine would also just be the traditional neuron setup that we've all heard of.
Yes.
That we all see.
Yeah. Yeah. Mm-hmm. Yeah, I think you can think of it that way. Yeah. And a hormone is by definition a signal that travels through the bloodstream to reach its target. Okay? So we mentioned that insulin has neuroactive properties, and when it acts as a neurotransmitter, we call it a neurotransmitter, but when insulin is released by the pancreas into the blood supply and then acts on muscle cells to influence the uptake of glucose, we call it a hormone, right? Same exact molecule, but it can be referred to as a neurotransmitter or a hormone based upon the method or the route by which it's traveling to reach its target. Okay? So amplification is increasing the potency of an original signal. And that can be done by several different mechanisms. And this is a nice example of three of them by activation of G proteins. We'll talk about G proteins today. Increasing production of a second messenger, in this case they mentioned cyclic AMP, or increasing the phosphorylation of proteins by increasing intracellular products that can carry out those downstream steps. Okay, so I wanted to talk about that before I talked about
communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the nerve impulse reaches the presynaptic axon terminal. Depolarization of the presynaptic membrane initiates the sequence of events leading to transmitter release and activation of receptors on the postsynaptic membrane. Shown here are the principal molecules and organelles necessary for release of neurotransmitter. Synaptic vesicles exist in distinct pools, either tethered to the cytoskeleton in a reserve pool or free in the cytoplasm. Some of the free vesicles make their way to the plasma membrane and dock as a series of priming reactions prepares the vesicular and plasma membranes for fusion. synaptic vesicles are drawn together via protein complexes, collectively called snares, that are expressed on the vesicle and presynaptic membrane. When the axon terminal is depolarized, voltage-gated calcium channels open, and calcium ions rush into the axon terminal. Some of the calcium ions bind to a protein on the synaptic vesicle membrane called synaptotagmin. When calcium binds to synaptotagmin on the synaptic vesicles nearest the active zone, the vesicles are drawn even closer to the presynaptic membrane. The vesicles fuse with the axon terminal membrane and release their transmitter cargo into the synaptic cleft. Some of the transmitter molecules bind to special receptor molecules in the postsynaptic membrane. The response of the postsynaptic cell, either excitation or inhibition, depends upon the particular neurotransmitter and receptor combination. For example, the receptor for acetylcholine is permeable to sodium. After binding acetylcholine, the channel opens and sodium ions enter the postsynaptic cell, thereby generating an excitatory postsynaptic response. Transmitters are inactivated or removed rapidly from the synaptic cleft so that transmission is brief and accurately follows the presynaptic input signal. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down acetylcholine into choline and acetate. The release of transmitter from the receptors causes the channels to close. Not all transmitters are broken down by enzymes in the synaptic cleft. Many transmitters are rapidly cleared from the synaptic cleft by being taken up into the presynaptic terminal by special proteins called transporters. This process is known as reuptake. Reuptake not only cuts off the synaptic activity promptly, but also allows the terminal to recycle transmitter molecules. The membrane needed for creating synaptic vesicles is also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are refilled with neurotransmitter molecules and are ready for another round of synaptic transmission.
So here you see, and the animation discusses these the animation discusses these specialized molecules that are called snares, which help the vesicle to dock and then fuse to the presynaptic membrane. Snares is a large, basically referred to a large family of proteins. These are all attachment receptors, attachment proteins, and they exist in many different mammalian cells. In neurons, we have both V snares, which refer to snares attached to the vesicle, and T snares, or snares attached to the target membrane. So in this instance, the target here refers to the target of the vesicle or the membrane of the presynaptic cell, right? So target snares fuse with V snares and help draw the vesicle, along with other molecules, help draw the vesicle toward the cell membrane so that it can fuse with the cell membrane and exocytose its neurotransmitter contents. Snares are also the targets of several different toxins, and this is an interesting one. The role of calcium in neuronal conduction. Calcium is a very essential, it's absolutely essential for maintenance of heart muscle contraction, smooth muscle contraction, and absolutely essential. We've talked a lot about sodium and potassium and chloride in the electrical workings of the nervous system, and we haven't mentioned much about calcium, but it is absolutely crucial in synaptic transmission. So here's how. Make sure I get the right one here. So calcium is necessary for the action of snares, both the V snares and the T snares, to interact with one another and then draw the vesicle toward the neuronal membrane. So it's involved in the function of the motor proteins that change conformation and allow the fusion of vesicles. Actin is involved. You might remember the microfilaments that are part of the cytoskeleton included 3 different types of filaments. There were neurofilaments, microtubules, and microfilaments, which were the same as actin. Okay, actin is a muscle protein. Actin and myosin are the 2 muscle proteins that slide against one another when a muscle contracts. And actin is the protein that's involved in drawing the vesicles closer to the presynaptic membrane. Calcium is important for muscle contraction. In the same way, calcium is necessary for the action of actin to draw the vesicle close to the presynaptic membrane. So this is a sort of a simple animation, but kind of gets the idea that there's a separate calcium channel in that presynaptic membrane. It's necessary, it's voltage gated, and it's necessary for that calcium channel to open before the vesicle can actually fuse and exocytose its contents. Not the voltage-gated sodium, and the sodium channels in the postsynaptic membrane, right? Oops, thank you. And maybe
by 2 broad families of receptor proteins, ionotropic and metabotropic receptors. Ionotropic receptors are multimers made up of at least 4 or 5 individual protein subunits, each of which contributes to the core of the ion channel. Ionotropic receptors contain 2 functional domains, an extracellular site that binds neurotransmitters, and a membrane-spanning domain that forms an ion channel. When a transmitter, or ligand, binds to an ionotropic receptor, the channel becomes activated and ions can flow through the channel. Ionotropic receptors generally mediate rapid postsynaptic effects. The postsynaptic potentials arise within a millisecond or 2 of an action potential invading the presynaptic terminal and last for only a few tens of milliseconds or less. The second family of neurotransmitter receptors are the metabotropic receptors, so called because the eventual movement of ions through a channel depends on 1 or more metabolic steps. Metabotropic receptors do not have ion channels as part of their structure. Instead, they affect other channels by the activation of intermediate molecules called G proteins. For this reason, metabotropic receptors are also called G-protein coupled receptors. Metabotropic receptors have an extracellular domain that contains the neurotransmitter binding site and an intracellular domain that binds to G proteins. The G protein shown here has 3 subunits, alpha, beta, and gamma. The alpha subunit binds to guanine nucleotides, either GTP or GDP. Binding of GDP allows the alpha subunit to bind to the beta and gamma subunits to form an inactive trimer. Binding of an extracellular signal activates the metabotropic receptor and causes GDP to be replaced with GTP on the alpha subunit of the G protein. When GTP binds, the G protein becomes activated and the alpha subunit dissociates from the beta-gamma complex. Following activation, both the GTP-bound alpha subunit and the free beta-gamma complex combine to downstream effector molecules that mediate a variety of responses in the target cell. For example, the G protein may interact directly with ion channels, causing the channels to open or close. Activated G proteins may also bind to other effector proteins, such as enzymes, that make intracellular messengers that mediate a variety of responses in the target cell. The activation of metabotropic receptors typically produces much slower responses, ranging from hundreds of milliseconds to minutes or even longer. A given transmitter may activate both ionotropic and metabotropic receptors to produce both fast and slow postsynaptic potentials at the same synapse.